Even when I was a little boy, I was fascinated by animal behavior. Then one day I realized that people actually made careers out of this. I got interested in bats for two reasons, really. First of all, they're very diverse animals, and as such they contribute to the diversity of the living world. And secondly, bats play an important role in ecosystems. Their behavior with their centralized provision strategy is similar to that of bees. And bees also play a key role in ecosystems as agents of pollination. All bees are good pollinators because they have branched hair on their body that helps pollen cling to it, ensuring the pollen gets from flower to flower. On this plot, within the framework of the European STEP project, we're studying how pollinators affect fruit production. Take these three kiwi fruit, for instance. The smallest one was pollinated by the wind, while the other two were pollinated by both wind and bees.
At Inra's Bees and the Environment unit in Avignon, one of the things we are studying is communities of wild bees. We get them ready for categorization by highlighting different morphological characteristics. Once their species have been determined, the bees are stored in bottles either for morphometric analysis later or to build upon our reference collection to help us identify these specimens.
Toward the end of the 1990s, beekeepers started noticing significant losses in their apiaries. They suspected that a certain number of bees were perishing outside of the apiary due to an accumulation of factors, be they climate change, the use of pesticides or disease. But at the time, there were no techniques available to address the problem. For some 10 years, my colleagues, especially here at Inra Avignon, teamed up with agricultural technical institutes to develop a system for tracking bees. It works like a barcode. A small electronic chip is attached to the insects' backs, so that we never lose track of them and can record every time they leave the hive to feed and come home to it. This was a major breakthrough in our research because it freed us from the constraints of the lab and allowed us to work with bees and their hives out in the field. Now we can follow their every move throughout the entire life cycle. Thanks to this kind of technology, we can now measure mortality rates in different cohorts, for example, and try to create models for population dynamics and determine what sources of environmental stress eventually lead to a decline in bee colonies. The study that we published last year in Science magazine had a major impact both at national and international level, because for the first time we were able to show that even at very low doses, pesticides can lead to indirect mortality with potential consequences for the dynamics of bee colonies. We're continuing in this direction by taking a broader approach and trying to decipher how and to what degree different environmental stress factors such as pesticides, climate change and disease come into play. For me, a researcher has to be creative and try to think outside the box. That requires a certain freedom of thought and freedom of movement in research.